Welcome, everyone. I know we still have people filing in, but I'd like to get started with today's seminar. This is our HMEI faculty seminar series. My name is Mike Celia. I'm the director of Princeton's High Meadows Environmental Institute, and it really is a pleasure to welcome you back, or maybe welcome you for the first time into our seminar series. This is a somewhat special seminar series in that all the talks are given by Princeton faculty members. We offer this seminar series for those who are new the first Tuesday of each month. So this is the first one of the semester in February, and we have the pleasure of hearing from Bryan Grenfell, whom I will introduce in just a minute. But just as a heads up, the first Tuesday in March, we'll hear from Ram Ramaswamy, who's the director of the Geophysical Fluid Dynamics Lab, also a professor in the program in Atmospheric and Oceanic Sciences here at Princeton. In April, we'll hear from our newest hire in HMEI, Professor Alison Carruth, who's jointly appointed between the Institute and the Program in American Studies. And in May, we'll hear from another one of our faculty members, Reed Maxwell, jointly appointed with us and Civil and Environmental Engineering. Just as a reminder, as we've done since we have gone to this remote format, everyone in the audience is invited to submit questions or comments. And to do that, you should use the Q&A box. The chat function is disabled for participants, but you can transmit your questions and comments through the Q&A box on your screen. You're welcome to do that anytime during the talk. And as a reminder, the seminar is being recorded. Okay, as I said, today's speaker, as you know, is Bryan Grenfell. And as we have done since we've gone remote, we will also have a discussant who will join the conversation at the end of Bryan's talk. And I'm really delighted that today's discussant for us is Professor Jessica Metcalf. I'll say a few words about Jess when we get to the end of Bryan's talk. All right. Bryan Grenfell is the Kathryn Briger and Sarah Fenton Professor of Ecology and Evolutionary Biology and Public Policy, jointly appointed in the Department of Ecology and Evolutionary Biology, EEB, and the School for Public and International Affairs, SPIA, here at Princeton. Bryan is a world-famous population ecologist whose work is at the interface of theoretical models and empirical data. He studies population dynamics of infectious diseases, and much of his work focuses on combinations of epidemiology, evolution, and control by vaccination. He's worked extensively on measles, as well as other diseases, including influenza and HIV. And more recently, as you can see from the title, he's been involved in numerous efforts related to COVID-19. Bryan has an incredible academic and publication record. He's received numerous prestigious awards, of which I will mention only a few because he asked me to keep this brief. So I picked out only a handful. He's received the Scientific Medal from the Zoological Society of London. He's elected as a Fellow of the Royal Society of London. He's been elected a Fellow of the American Association for the Advancement of Science. He's received an honorary doctorate from Sheffield University, and he's been elected a member of the American Academy of Arts and Science. Bryan's been a terrific contributor to our institute, HMEI, including teaching an important course on infectious disease. And it really is a great pleasure for me to introduce Bryan and to thank him for taking time to give this talk, because I know he and Jess both are incredibly busy dealing with many issues related to COVID-19. So please join me in giving a very big virtual welcome to our great friend, Professor Bryan Grenfell. Bryan, the screen is yours.
Great, thank you, Mike, and thanks. Welcome to everyone, and thank you very much. This is such a great seminar series, and it's a real pleasure to give a talk. I'm just going to share my screen. Always takes a second to get this working. Can somebody confirm that they can see the screen?
Yes, can see it fine.
Great, thank you. So I want to give quite a brief talk today, which will just open us up for more discussion about COVID-19, a lot of activity in our community in Princeton and hugely beyond that on all scientific aspects of the outbreak, and science has been incredibly great in all fields and stepping up to the plate here, I think. I want to step back and root this talk in work that our groups have done in the past on endemic infections that are around all the time, like measles, influenza, the current coronaviruses, and see how they inform our understanding or perhaps limits of our understanding of COVID-19. I want to focus on population dynamics and immunoepidemiology. I want to talk about the evolution of immune escape, which is a huge area and a growing area. And I want to talk about the interaction of COVID directly and indirectly with lots of other infections. I was going to do this from work and inflict my terrible blackboard technique on you. Instead, I'm at home and I'm going to inflict my even more terrible drawing skills with a mouse. Many people over decades were involved in this work, and very kind funding from these funders for a lot of this work. I would have had to go in a year ago, I'd have had to go into more details, or maybe 15 months ago, about what the basic parameters of epidemic spread are. Now everyone knows what R0 is, what herd immunity is, hugely widespread. So I don't need to go into much detail, but just to give you a quick feel, if we assume R0 is 5 at the start of a new epidemic here, so everybody is susceptible, then each person will infect 5 other people, but that will cause obviously then 25 people and so on. That'll cause an exponential increase in the number of cases. But if the infection is completely immunizing, if there's no retransmission of any reacquired infection after you've had it the first time, then that's going to deplete the susceptibles very quickly. So then the number of people you can infect is reduced, because a lot of the time you're hitting immune people. This is at the peak where each infection causes 1 more, and the effective reproduction ratio drops. Obviously, non-pharmaceutical interventions are going to do that a lot for COVID, and I'll come back to that. So this causes really indirect protection. So this person is indirectly protected, for example, and that leads to this idea of herd immunity. So the epidemic dies away if it's an immunizing infection. That's a big if. And when it's spread through the population. And in fact, that level of susceptibility would be how much you would need to vaccinate in this very simple system. There are many decades of theoretical work in that area. So you're probably very familiar with all that stuff. And the basic extension of that to endemic infections is we really need to think about where new susceptibles come from, and the details here don't matter, but they come from births effectively. Here's the transmission rate, and the transmission rate is often seasonally driven. Stochasticity is important as well. So this is effectively a simple pendulum, and it's a forced pendulum in the endemic state. Now, what do I mean by the endemic state? So this is when births and demography is involved as well. So this is a lovely paper last year by wonderful research associate in HEMI, led by Rachel Baker, in Science, where she looked at the effect of seasonality in transmission. It's by analogy with other coronaviruses. COVID should certainly be a winter disease, and posited at the time that the huge initial pandemic. So here's the pandemic, and here's a pandemic with non-pharmaceutical interventions at the time. The huge pandemic would be such an oversupply of susceptibles that you wouldn't see strong seasonal effects. But then by endemic, this system, this is assuming it's pretty strongly immunizing, would drop down to these endemic cycles. And what I'm really going to do now is to ask what those endemic cycles come back to and tell us about the uncertainties of COVID. So when we look at endemic dynamics of acute immunizing infections, just a little bit of background here, and look at pre-vaccination dynamics in large cities, the susceptibles, as I say, topped up births, and there's seasonality in transmission. So it's this forced oscillator, and we tend to see cycles. Here there were annual cycles winding out beautifully to biennial cycles, and models capture these things reasonably well. And this is in London, and essentially, if the birth rate was very high in the baby boom here in 1947 peaked, then the susceptibles top up more quickly, so you get annual epidemics. And then when birth rates drop, it winds out into these lovely biennial patterns. So the annual to biennial is because the susceptible supply tunes the dynamics as well as the seasonality. So if we put those two things together, we can draw a bifurcation diagram. So each point on this graph is a particular birth rate and strength of seasonal amplitude of seasonal forcing, and then you run the model, the resultant model forward, and you see what the long-term dynamics are. And when seasonality is very low, it's not a very strongly forced epidemic, so you would tend to get annual in blue or biennial dynamics. London, for example, in the baby boom, birth rate modifies this. So when birth rate's high, you get annual dynamics, then you get biennial dynamics. In very strongly forced system, there was a big vote to look for chaos in measles 20 or 30 years ago. In fact, we do see something like that in very strongly forced systems, and this is wonderful work by Matt Ferrari some years ago, where strong seasonal forcing causes very violently irregular dynamics. And then as an intermediate, and this is lovely work by Ben Diel, who's now at Oregon State, even historical data for US cities, because school semesters were a bit longer, measles, climatic drivers probably aren't that important compared to crowding because it's so transmissible. You see these chaotic-like patterns, but seasonality and birth rate tune the dynamics. Here's an example, just logging the cases here for measles in the UK through time, and a wavelet spectrum here shows that you get this predominant 2-year cycle, and then as they cranked up vaccination slowly in the UK, and you get this lovely increase in the period because it takes longer to top up the susceptibles. And all this is coming to, so we're going to come to COVID in a second, all this is coming to saying that evidence for strong herd immunity, circumstantial dynamical evidence, would be multi-annual cycles tuned by susceptible recruitment. So you get these multi-annual cycles because the number of susceptibles needs to be topped up, and susceptible recruitment tunes that. So we're going to come back to that and evaluate that in the context of COVID. So COVID-19 dynamics, again, if you follow this, it's very well covered in New York Times, Washington Post, lots of other places. Super spreaders are extremely important. Age of particularly disease is extremely important. Space and stochasticity, the huge impact of non-pharmaceutical interventions and social forces, vaccination that's coming up, and so on. The other things I'm going to we've done a bunch of work on these things, but I'm going to concentrate on other things in the rest of this talk. The strength of clinical and transmission blocking immunity, and I'm really going to focus on transmission blocking immunity here. If you get immune for one reason or another, immunity or natural immunity or vaccination, do you retransmit it if you get it again? That's an absolutely central question for understanding the pandemic at the moment. And I'm going to talk about evolutionary issues, and I'm going to touch on pathogen community dynamics at the end. So now we're going to move from the SIR model, and we're going to think about a more imperfectly immunizing infection. So let's make a toy model. The simplest assumption, and I apologize profusely for my incompetence at drawing with a mouse here. This bit's better on the blackboard. the simplest assumption, the strongest assumption is that everyone is immune for a year or two, let's say, or whatever period, and then goes back to a completely susceptible class. Well, if you imagine that that process is a little bit averaged, it depends on the waiting times that you're immune for, obviously, then this is like an enormous birth rate. So this should produce incredibly annual dynamics. It should get rid of all those multi-annual patterns. So indeed, if you imagine, this is birth rate, if you imagine a baby boom and a completely immunizing SIR infection, then the baby boom, you'll get high frequency oscillations, and then when birth rates drop, you'll get multi-annual oscillations. So that would be our signature that there's a strong immune effect. For completely SIRS dynamics, then the epidemics just sail through the high birth rate, so you don't get that effect. Vaccination does this as well. That's very important. Imagine a drop in birth rate. That's to first order a little bit like an increase in vaccination rate. So simply giving a single shot of vaccine, if you have a tremendously strong loss of immunity, you have to really revaccinate to have a strong effect. And this idea of the jump from SIR to SIRS was coined by ex-grad student Sinead Morris in this paper, and she called it demographic buffering. So let me give an example of, go back to some specific biology of a complicated infection with very imperfect immunity. Not COVID yet, but we're getting there. But it's very important to look at these particular cases. This is rotavirus, major diarrheal virus. Before vaccine used to kill perhaps 500,000 infants a year around the world. Very good vaccines were produced, developed starting a couple of decades ago. And this is lovely work by actually EEB alum and former postdoc and now a professor at Yale, Jenny Pitzer. And immunity only lasts about a year for rotavirus, but you get a sort of a primary set of infections, right? And then you get susceptible again, and then secondary, and then a third set of infections. And the key observation is all the action of transmission really happens in this primary infection. Young infants get this infection, and it's incredibly transmissible, much more than measles. Has an R0 of about 30 or 40 in this sequence. So I didn't expect, because immunity is so imperfect, you'd see any of those tracking effects of demography. Ginny, however, disagreed with me, and she looked at birth rate against the timing of epidemics in the US and found that, in fact, if you have a high birth rate, the annual epidemic of rotavirus started rather early. And you can read the science paper to get at the details here, but she was able to successfully make a model of the pre-vaccination or the early vaccination period, and then was able to predict that you'd have a strong delay here, and that's a sign of herd immunity, because you're getting essentially, you're replacing those primary infections with vaccinees. So even in perfect immunity, the devil is in the detail. Just as another nice aside, in this paper in 2009, Ginny projected forward, and indeed, when you vaccinate very well, the US vaccinates at high levels. There's vaccines are more imperfect in lower middle income countries, which is a significant problem. Then you will actually get the emergence of biennial dynamics, and indeed we've seen that. So you see this as Ginny's work, a many years ahead prediction of biennial dynamics. So even though immunity lasts about a year, because of that complication of the biology, then you get strong immune protection. So Sinead Morris in her paper developed therefore a model with an intermediate tunable level of immunity, and it's all in this. Let's concentrate on this parameter epsilon. So you go through susceptible, infected, and recovered, but then you come back not to a fully susceptible class, but to a partially susceptible class, and then these parameters alpha and epsilon, and let's focus on epsilon, concentrate the amount of susceptibility and the amount of transmission. So if alpha and epsilon are both 0, you end up with our old friend, the SIR model. If they're both 1, you end up with SIRS. And let's focus on epsilon. The larger epsilon is, the larger the amount of secondary transmission, the more worried we are. And this is lovely work published last year and led by grad student in QCB, Shadi Sadroy, and Caroline Wagner, who was a postdoc and is now a professor of mechanical engineering at McGill. And what they did was look at for weak immunity and strong immunity. And for COVID, we still don't have a strong field, because to measure immunity, you imagine, is very hard, because you really have to follow people and see how much they infected their contacts if you want to measure transmission blocking immunity. If you have strong immunity, you get these, and this was looking forward from the start of the epidemic. You can easily damp down the infection, and the proportion of people in different immune classes is shown here. And this is without vaccination on the top row. If you have weak immunity, you would have much more activity. You'd have much more frequent epidemics than in this case, and you would have this big buildup of partially immune people here. And vaccination, you get roughly the same patterns. And again, the retransmission following vaccination at the time is very important. A lot of work since, some evidence that there's a degree of immunity, plus looking at the endemic coronaviruses, but many uncertainties. However, two complications have come up recently, which is quite understandably, a lot of public health folks would like to, because of shortages, global shortages really of the vaccine until they really ramp up, they wanted to increase vaccine dose spacing. So there's an optimal, the trials indicate an optimal spacing of 3 weeks or a month between doses. Could we give a much wider spacing? The UK and Canada are doing this, for example, particularly the UK, and therefore infect more people. So in a paper that's just about to go into MedArt archive and be submitted, and again, Shadi and Caroline did a fantastic job. And this has got lots of boxes and arrows, but it's about the simplest model one could make. It ignores heterogeneity, space, and so on. And if you read Shadi and Caroline's paper, they explore that if you're interested in detail. Essentially, we're adding two vaccine classes where you can separate people who are vaccinated once and vaccinated twice, and therefore look at all the sort of partial infected and susceptible people. And the bottom line here, and I've taken the pessimistic case here of rather imperfect immunity just to illustrate it. Then if we just look at one dose, in fact, it would be widely spaced doses, but just as an extreme, let's take up one dose versus closely spaced doses with relatively imperfect immunity, then indeed, if you compare that epidemic with that one, you really do better. And here we're ramping it up. If you could ramp it up quicker, you do even better. Stretching the doses is good epidemiologically in the short term, but if immunity is imperfect, as in this case, then compared with giving a slightly stronger dose of immunity with two doses, if that's the way the biology is set up in the model here, then you see you get this epidemic, and that's much smaller in this case. So you do well in the short term, but if immunity is strong, which I'm not showing here, you're fine with one dose. But if immunity is weak, then it's a much weaker effect. So we also see, and in particular, these are the people who are susceptible, who've gone back to being susceptible after one dose of the vaccine. You see those are building up. So there's a big buildup of partially immune individuals. Could that cause a selective pressure for the evolution of immune escape? So let me illustrate what immune escape is. So this recalls some earlier work we did on very simple models of this, and this is with Eddie Holmes and Ollie Pybus back in the day. If we briefly compare measles and seasonal influenza, so they're both RNA viruses, they've both got a high error rate, and this is a cartoon. These are supposed to be different. These are supposed to be different strains, which mutation, just as a cartoon. However, measles has incredibly strong cross immunity. There's no immune escape at all with measles. Therefore, the phylogeny of the surface molecules is quite boring. There are no strong, there are spatial patterns, but no really strong patterns. And you get these typical SR dynamics. Influenza, as we all know, you're immune somewhat against it. Just to give a crude cartoon here, you're immune against this year's strain, but then next year or at some point later on, the virus will evolve and you'll become susceptible again. So here you have this lovely ladder-like phylogeny with replacement of the next fit strain that can get around population immunity takes over and so on, and you get this ladder-like phylogeny. So we made a very, very simple conceptual model here. Imagine that you've got immune pressure, and you plot just in a conceptual way the strength of selection. So if the immune pressure is really strong, then the strength of selection is very high, but the viral abundance is very low. You've just clobbered the infection. Your immune system clobbers the infection, the secondary infection, so you never retransmit it. And obviously viral abundance is proportional to transmission. So here there's strong selection, weak transmission, so there's not much evolutionary novelty transmitted. Here there's strong transmission because you're not controlling it, but quite weak selection. So some intermediate level of partial immunity would be when crudely speaking, the immune novelty would come up. So in a paper that's just the same paper that's just submitted now, complicated outcomes. So here's that trade-off graph, and Shadi and Caroline looked at lots of options. There isn't strong selection in primary immune people from the natural infection, or 1 or 2 dose immunity. Here, 1 dose is worse in terms of selection. Here, selection is bad in all cases. And all I want you to take away here is these are the different sorts of secondary infected people. We can't model immunity explicitly here. There are too many unknowns, but you would tend to see bursts in, and this is assuming an outbreak in a single city for simplicity. You would see, if immunity is weak here, you would see bursts of potential this denominator rising, where you would expect to see the origins of immune escape. And of course, we've already seen a lot of evolution from COVID, even when numbers of immune people were very low, for lots of interesting reasons we can go into. And exactly which of these assumptions dictates whether it's the primary infection or 1 dose or 2 dose that does that. A really important point here is that this looks as though the threat only happens sometimes, but if you imagine all the countries, all the cities in the world and the hinterland with different levels of vaccination, different levels of seasonality and so on, we really need to get high global vaccination rates, morally, obviously, and in terms of public health, but also, and many people have said this, to limit the evolutionary potential of the virus. So let's come back to that immune tracking, the dynamics tracking immunity idea. And this is lovely work ongoing by Daniel Park. And Daniel's been using data very kindly given by the BioFire company, and they have for example, a respiratory virus panel that lots of hospitals use it, and they've given us the anonymized data to look at the incidence of lots of infections. And this echoes lovely work by Stephen Kistler and colleagues on endemic coronaviruses here in published last year. And if you just look at 2 of the 4 endemic strains of coronaviruses here, HKU1 and OC43, as Stephen Kistler pointed out, they're out of phase. So that indicates that there may be some competition between them, some cross immunity. And Daniel's interested in whether these biennial patterns are caused by homologous immunity or within the given strains, or interactions between them, and that is very early in that stage. But what about the epidemiological impact of birth rate? Now, we only have a few years data here, but we have lots of regions of the US, and very preliminary, just looking at the center of gravity of the epidemic, which for rotavirus would show a trend pre-vaccination. We don't see any trend. So this implies that the immunity is relatively weak for these coronaviruses. Very early days, not a full correction for effects of seasonality and so on, but we see that preliminary effect in the data. And then finally, I just want to touch on the polymicrobial bit, which is, there's been a huge natural experiment here. We've all seen that if you're in lockdown and so on, you're not getting any other infections. And this is, again, lovely work by Rachel and by Daniel Park, showing that everything else, many other infections compared to normal years, which is the curve here. This is RSV, respiratory syncytial virus, which is a very important virus of young children. Really, things have fallen through the floor. And in a recent PNAS paper, Rachel and Daniel, the trouble is, it sounds great that the epidemic has declined a lot, but we know from in an SIR world that means the susceptibles are going to build up, right? And therefore, we do expect, and this is one scenario, we're not sure what's going to happen yet. We expect the epidemics to come back and to come back as very large epidemics in the future, spread over the next few years. And the devil, again, really is in the detail of when that's going to happen. Also, this happens for influenza, and there isn't an RSV vaccine currently, but deployment of vaccines optimally would be a kind of interesting way to get at this. These perturbations will also give us, hopefully, much more understanding about the epidemiology of the infections. Something I've not touched on here, but we're very interested in our group and with Jess's group is, and this is lovely work by Lojin Yang and Daniel again, is the effect of immune waning and boosting on this, which is a very important component. So that was just a quick whiz around, just a small piece of what the community and in our local small way we've been doing on COVID, and lessons and also unexplained things from lots of other infections and endemic infections. Just to say, we in the broader community, a lot of other work has been done, from very local applied things to the seasonality questions, and to, for example, led by Lojin Yang and folks from Simon Levin's group, the social dynamics of mask wearing and vaccination and so on. A really interesting one, just finally, is led by former grad student of Simon's, Dylan Morris, who worked with a lot of biophysicists, looking at the mechanisms that underlie the seasonality of a lot of these viruses in droplets and so on, respiratory viruses. And that's an area that has been looked at heuristically and phenomenologically for a long time, but not mechanistically. And getting in the engineers and the biophysicists for theory, but also for empirical understanding has been extremely important. And I think that's a really great way forward for our field. So I'm going to knock off there, and hopefully I've maximized the amount of time we have for questions.
It's fantastic. Thank you, Brian. So we will now go into the discussion part, and as our discussant for today's talk, we have Professor Jessica Metcalf, as I already mentioned. Very briefly, Jess is also jointly appointed as a faculty member between EEB and SPIA. She's a demographer with broad interests in evolutionary ecology, infectious disease dynamics, and public policy. She serves, among other things, as the co-director of the Global Health Program here at Princeton, and she also has been a major contributor to Princeton's ongoing COVID-19 efforts. So, Jess, I'll turn it over to you.
Thanks very much, Michael. It's great to be here. A lovely talk as ever, Brian. So I thought, just to set us off, it might be useful to ask, what do you think this pandemic means for the future of our field?
So that's a great question. I mean, I think, and we always have great interactions with our fantastic colleague at Gabe Vecchi about this, of course, comparing it as a kind of meteorology and climate science as a kind of control. And I think we've learned from this that you've got to really think big. I think we need really to be modeling and understanding biologically a lot more about systems immunology and a lot more about the population dynamics of infectious diseases. We need a case law of infectious disease dynamics, essentially, because we don't know what the next, we have some idea, but we don't really know what the next threat is. But the theory's all very well, theory's fine, but really, as I alluded to with the stuff Dylan was involved in, and Logan, reaching out to biophysics and engineering or to social science, is that we really need better data. And I know you've been thinking about this in terms of immunology. What do we need to know immunologically, and where would you go from there?
Well, I think that we've been thinking about this together, right, Brian? One of the really fascinating questions that will allow us to better understand what prevents pathogens from spreading or prevents them from emerging will be rooted in what our own immune systems are doing, since we are, in some sense, the raw material amongst which these pathogens must spread, and which is captured by those SIR and SIRS continuum of models you point out. So I think what would be wonderful would be a global immunological observatory where we collate data from people all around the world, and you would necessarily need a global perspective, because if a pathogen's anywhere, everywhere is a threat, to understand what people's immune responses are everywhere, which would both give us potentially early warning for pathogen emergence, but also a sense of the substrate, I know you hate that word, across which pathogens could potentially spread.
And it would also give us, a wonderful material to look at autoimmunity, which is a huge problem, to look at cancer immunity. It would be hugely important, I think.
Yeah, systems immunology seems like one of the big next frontiers. And of course, as you said, theory is all very well, but you need data to underpin the theory and to push us forward. This links a little to the question of immunology and what we know about the immune system. A couple of questions have come up in the chat, and do keep them coming, everybody. People are very curious. So Sabrina Smith asks, initially the framing seemed to be that mutations would be slow and insignificant, but now we're seeing all these problematic mutations cropping up all over the world. And so do you have thoughts about why that's what that means for what we understand of the evolutionary potential? And does this have implications for this pathogen becoming endemic?
Let me speculate, and this is wild and dangerous speculation because I'm not a molecular biologist, and the molecular biologist, please, on the line, and the structural biologist jumped down my throat. But it's interesting to compare it, I think, and you're absolutely right that that this I think wasn't expected at the start because there's some error correction in the coronaviruses and so on. But if you compare it with flu, so flu gets into cells with a hemagglutinin receptor, and then it gets out. It has to cut that sugar link with neuraminidase, another surface protein, to get out, so that there's a balance there to some extent. I wonder if it's about, and you obviously have to have the plasticity, the spike protein gloms onto this very, very important physiological molecule that we have, the ACE2 receptor. And the harder it gloms, the better it seems to do, which is, I think, what these various variants are doing. And I don't know what the upper limit of that is yet. So maybe the compensate, unless you have so many mutations, you completely muck up the spike, then how many mutations can you add? And it's really a structural biology, virology question. And the more we all talk at different scales, the better, I think. But I wonder if it's because of that, but that's complete speculation.
So I'm going to say it again just to make sure I understood. The ACE2 receptor is both how the virus gets into the cell and how our immune system sees it. So it has wiggle room because it seems to be able to get into the cell quite easily and under slightly different conformations.
So with flu, my understanding, and we have two epidemiologists talking
both
about
at the edges of
virology here. So that with flu, the better, if you have a really, really sticky hemagglutinin that pushes you onto the receptor, you have to cut that same link with another molecule, neuraminidase. That sort of duality mechanism doesn't apply to the coronavirus. So it's only that the more you can glom onto cells and push antibodies out of the way, maybe the better you do. There's got to be an upper limit on that, and I know the cryo-EM folks are working crazily hard on this to get at it. But maybe someone else in the chat actually knows what they're talking about here.
It's true. It does look like there's a lot of great questions. And then I think one of the things we conflated a bit there, or at least I conflated, was that there's going to be selection on different aspects of what the virus can do. Selection for greater transmission, which might be about this glomming on, but it also might be a piece to do with immunity. So there's all sorts of pieces that the virus can play with. James Koopman has, sorry.
So there's one more quick thing, and I don't know if Dylan's on. We probably don't know yet what about the genome of the virus controls its ability to persist under different environmental conditions. That's another potential axis of variation. Anyway, carry on with the question from Jim.
Absolutely. So it has just like everything else we think about, it has a phenotype in some sense, and there's all sorts of angles to it. So James Koopman asked a question, which I think is asking about the kind of evolvability of the virus. So the RDRP of SARS-CoV-2 occupies 2/3 of its genome, and it's presumably been tweaked to make it just so. So how can we counter this ability of the virus to adapt? So he's sort of asking whether global vaccination will be enough, given that the virus has so much potential wiggle room.
So that's, as ever, a great question. I think let's be optimistic and assume that the only thing that's varying is the makeup of the spike. There may be other things involved. Then, could this absolute triumph of the mRNA vaccines, could the structural biologists work out what the constellation of mutations is, and therefore could you have a cocktail of spike that allows you to cover the bases? Now, my expert friends worry about that because you don't want to crowd too many things. There may be downsides, there may be immunodominance problems, and so on. But I think, and this isn't going to be that quick that you can make an individual one of these. But are there yet more vaccine things that can be done? Now, the other point about the spike is if you could make a therapeutic that exploited some of these characteristics, then maybe therapeutics has got to be an important thing to ramp up.
And just to translate, so immunodominance sort of means our immune system gets obsessed with the wrong thing and doesn't loses the ability
that's
to
right.
learn.
Or original antigenic sin. So you get the infection with the wild type, and then you get the mutant, and your immune system, like with flu, remembers the wild type immunity, which is not great.
Yeah, well, it's not great, but it is very interesting. Nina Masters asks, so she works on vaccine preventable diseases, and she was wondering about these mandatory pediatric vaccines. So what do you think might happen in the next year or so, not just with flu and RSV, but other pediatric infections for which infections have sure been falling, but so have vaccination rates in many settings?
Yeah, I mean, and I think it's something we've discussed with measles, I think, I mean, again, a lot of complexity of the biology, but the one thing you can say is if susceptibles build up with those infections, if there are more primary susceptibles because kids missed the vaccine, missed the epidemic, they're gonna get the infection at some point. Now, our colleagues, Jess, who are expert with RSV, say if children get it a bit later, maybe that's not as bad, but it'll again depend a lot on the age, the disease profile with age, for example, if you get it older, because if you don't get it now, when it does come roaring back, the kids who at the quotes right age will get it, but the older folks, the older children will get it as well. So it really depends on those sorts of age profiles of susceptibility. But yes, I think it's a tremendous worry, and I would have expected to see more big measles epidemics by now. I don't know about you.
Yeah, no, absolutely. I mean, I've been working to get data on Madagascar to try and understand how the vaccination rates have fallen there. And they did just have a large outbreak. So they unfortunately got immunity the wrong way quite recently, but I think it is a concern. And I know that WHO and other organizations are worrying about this. There is a question related to this sort of age profile of immunity and how it plays out, which is what does our research mean for the future for people with serious autoimmune diseases?
So I think, I mean, I think the vaccines have not been tested in those populations, and obviously specific clinical advice is the key thing. I mean, I guess knowing what the advice is about whether you should take influenza vaccine is a kind of a guide here, really. But you know, it obviously, if despite all the caveats about immunity being maybe being imperfect, if if the COVID vaccines give some clinical protection, that's very important. So that's an important thing to weigh up.
And to take a million steps back, one of the things about the Global Immunological Observatory that Bryan and I are so excited about is that it should provide us with samples of people's immune systems as they grow older and as they develop different profiles, and people in different contexts with different exposures. Apparently 80% of autoimmune diseases are concentrated in women, and I'm very fascinated by that particular angle on differences in immunity. So hopefully, this isn't by no means an immediate solution, but hopefully the more we know about how systems immunology works, the better we'll be placed to at least understand what drives these sorts of outcomes. So Maya Chung asks, what causes a disease to mutate? Is it just population-based, or might there be influences like temperature or climate?
So, certainly, and it's something we touched on in the previous discussion. Actually, if there were, for example, drivers in the viral genome that meant that you could select for better tolerance of bad conditions, for example, high humidity, in terms of the viral envelope and so on, then you could certainly see those effects. I think we don't understand the mechanisms of environmental drivers on, for example, respiratory virus yet. And I think that's that's a gap. And hopefully, you know, one small positive thing of the pandemic is there'll be a much more active area of research. But I think that's a really great question, actually. We concentrate on the immunity and the spike, but environmental drivers could be very important as well.
And I think our friend and colleague Eddie Holmes would remind us at this stage that, of course, mutation is always happening, yeah. But of course, as a background rate. But you're absolutely right that the speed what that rate is, yeah, might be temperature dependent, might be replication dependent. There's all sorts of ways in which these things still need to be unpicked. Yeah. So Emily Tseng is curious about our perspective on how big tech companies have responded to the clear and urgent need for better empirical data and global observatories. Are there trade-offs with privacy, data quality, etc.? Are modelers getting the data that they need?
So I think, is the answer to the last one no, Jess?
Well, I think Bryan is famously greedy. He always wants more data.
I mean, I think the answer is I think the answer is no. But I mean, that's because it wasn't a priority, and that's nobody's fault, right? I think those are really great questions, and they're questions we've agonized about. And there's an eLife paper where we explore some of these ideas. I think the question of whether bits of this are commercialized, the full observatory would be a huge thing, I think. Commercialized, particularly in endemic countries for lots of important infections, you really need to empower people. Could you then persuade people that they could use, our colleague Michael Min has had great thoughts about this, they could have a kit to do the immunology, but then they might put up very anonymized data on immune status against different diseases by age class into some repository. I think there are lots of plans and a lot of things that need to be done, but certainly those questions are things we agonize about.
Yeah, just echoing your emphasis on privacy and data quality are really important. I feel like part of the question that Emily was asking also was something you and I have touched on, Bryan, which is big tech companies. So data like the Google data on mobility and those other threads. And those, I mean, it has been, I think there's a glass half empty, glass half full perspective, because it is extraordinary how much information has been available on how people move during this pandemic. Yeah. again, all the concerns about the ethics around that are really important and don't go away. But and who gets access to that data also remains a question. Do you have other thoughts, Brian?
No, I mean, I think you've nailed it. I mean, you know, somewhere in the intersection of that and, you know, 23andMe and the genomics companies. But, you know, if there's a sort of an open data for fending pandemic sweet spot there, where you can get anonymized data, because, you know, if we were looking at something like measles, we're talking about proportion of that measles is the simple one, proportion of immune people by age in broad age classes through time, you know, annually. We're not talking about, because you don't need weekly data or anything for these immune measures often, because they're a sort of an integral of past cases. So you only need them at broader intervals. So if you could just get those sorts of data much more broadly than currently, and I think there are many paths to getting those data, but you know, I think big tech could certainly play a part in this, but there's a lot that needs to be navigated, as with the mobility stuff you talked about, Jess.
And of course, just basic public health, right? I mean, there's such a shocking paucity of testing for so long. We had such a sort of strange window onto this pandemic because the I-box in Bryan's SIR models was very incompletely specified, let alone the S-box, which is what Bryan and I are sort of talking about when we talk about a global immunological observatory.
And very few people did sequencing, actually.
And still people are sort of playing catch up on sequencing. And that's something I think worth remembering as well, that one would try and be very careful not to use the terms UK variant and South African variant, because what that actually probably reflects is that these countries are quite good at sequencing, yes. More than that they popped up there. So Kathryn Zandanella asks, what is it about SARS-CoV-2 that leads to the lack of seasonality that we've seen so far? Is it just that this pandemic is new and that it will become seasonal?
So I think that last point is a good way to put it. I mean, we're channeling Rachel's work here, which also one saw with 1918 and other pandemics, actually. So if they're strongly seasonal, right? But the transmission rate is the proportion of susceptibles times this seasonally varying transmission rate. So if there's a huge proportion of susceptibles, that'll really drive things early on. Rachel's since gone on to show very subtle interactions with non-pharmaceutical interventions. So if they keep the transmission rate a lid on it, then as the epidemic goes on, seasonal effects will happen. And there's a lot more to be done, and no final words yet, but you know, getting a mechanism and getting more data has got to be the way to address this.
Laurie Abrams asks, can you comment on the Swedish inverted commas experiment? Why do we think we didn't see more evolution of the virus given the unchecked transmission there? And I think part of the answer there is just what you're looking for, right? You won't see new variants unless you're sequencing a bunch of things.
Yes.
But the other part is I don't think we really have pinned down, you know, certainly more circulating virus gives you more opportunities for escape, but there's a bit more to it than that, right, Brian? That there is at least speculation about the context that favor. Yeah,
so you need the new variants to pop up in individuals. Again, the model I showed was extremely crude. The absolute zero order model. Within host dynamics will matter. The timing of different bits of the immune system will matter. So it's the mutation rate times how much selection your immune system gives, and then do you happen to transmit it, which might be a rare event. But I think unless you've got the sequence information, then we can't really know.
And I would be greedy and add, you know, we need the sequence information, and then we need to start thinking about the immunology and trying to understand how our immune systems might be chasing these viruses around. And we can't do that unless we have historical samples as well as contemporary samples.
Exactly.
So we need a global immunological observatory, one might say.
Exactly.
So Rob Sprinkle asks, do you expect covariants to turn COVID into a disease requiring vaccine variants every year? And assuming that it keeps varying and keeps returning, and with many kids likely to get measles in coming years, and since measles wipes out pre-existing immunity, as per Mina et al., should we expect that we have many more susceptibles than we now anticipate? That's a very pessimistic question.
So I think we concluded, and Mike Minner and you and I just, that strong ablation of previous immunity. So measles is a predator of immune cells and memory cells in the immune system. and therefore it knocks down for a period immunity against other infections. I think we decided that it was especially severe measles, maybe where you're hospitalized, where you'd get a strong effect like that. So the sort of cross-herd immunity effects, which I think among us I was pushing at the start, I'm not sure we see those now, because we don't see those interactions with pertussis. Annual vaccination, I don't think we know yet. I mean, I think it's strangely, though we seem to have been in this world forever. It's early days, I don't think we know yet. I would be very surprised if you wouldn't have frequent vaccinations, but we don't know. I would love to know which, or if any, of the current endemic coronaviruses were severe pandemics early on. I think that would be a really fascinating thing to know. Though COV2 is unique in its biological characteristics.
To echo the model that Bryan presented, everything sort of hinges on what secondary infections following either infection or vaccination look like. And we don't really know that yet for SARS-CoV-2 itself or for variant strains. So that, once we start pulling up data on that, we'll have a better sense, I think.
I hate to jump in, but I'm looking at the time. I know that both Bryan and Jess have engagements at 1:30. So I don't know if either of you or each of you would want to just give one final quick comment. I know we have other questions that we're not going to get to, but these have been terrific questions. So I will let each of you just make maybe a final quick comment, and then we'll try to wrap up so that we can keep on schedule.
So there's a last question I might tackle from Palevi Nuka, who asks about parts of Africa and Asia seeming to have lower COVID infection rates. Asia is, is very interesting, and I think has really responded. Many countries there have responded uniquely. For sub-Saharan Africa, Benjamin Rice, who's a brilliant postdoc in our groups, just led an enormous international team to suggest that it'd be very hard to know what's going on given current data repositories, and it's also an enormous simplification to crunch that entire continent down to a single outcome. There's probably very many trajectories going on in very many places. Bryan.
I'm just going to really echo Ben's stuff and say, universal, rapid dissemination of vaccines, tests, and so on around the world is absolutely key.
Excellent. Well, Bryan, thank you very much. Jess, thank you as well. Terrific presentation and fantastic discussion. Thanks everyone for sending in questions. And thanks all of you who are in the audience. I'll remind you that we will have our second talk of the semester, the first Tuesday of next month, which is March. It will be by our friend Ram Ramaswamy, Director of the Geophysical Fluid Dynamics Lab, GFDL, and jointly appointed as a faculty member in the Atmospheric and Oceanic Sciences program here. Looking forward to seeing you then. Thank you all very much. Stay safe and be well. Thank you again, Bryan and Jess.